Hello, my name is Rahul Roy, and I'm a new assistant professor in the Department of Biology at St. Catherine University. I was a US Department of Agricultural Research Fellow at the University of Minnesota Twin Cities before this, and I have been working on the biology of nectar production in flowers and the secrets it holds. Nectar is, in a simple sense, a sugary reward that is produced by flowers that attracts pollinators and promotes cross pollination. So you can see here a drop of nectar produced by an Arabidopsis flower. Um, Pollinators usually go for the nectar and in the process brush against pollen, which then can be transferred on to the pistil or the female sex organs of a flower, of a different flower, and cause cross pollination and fruit set. We also know that nectar and pollen together are converted into honey, which we are so fond of. Now, this is a very economically important phenomena because species such as oilseeds, almonds, blueberries, apples, pumpkins, cranberries, and many other crops depend on pollinator services for a proper fruit set. This amounts to almost a $20 billion market share. Cross-pollination due to attraction by nectar and pollen is also important for proper fruit set, as we see here, strawberry that has been pollinated by bees looks more like a strawberry, whereas the self-pollinated strawberries look more defective and have not found a proper fruit. Now, nectar is produced by structures known as nectaries that are at the base of the flowers, and this is something that my research has been investigating. So you can see here the nectary in a squash or a pumpkin flower is cup-shaped and produces the nectar into this hollow cavity here, whereas in Arabidopsis it can be like a bean-shaped structure which produces nectar. Nectaries are very interesting structures because they are under a very complex hormonal and circadian control. They just don't ooze out nectar. They produce nectar at a very specific time of the day. And we are trying to understand the genetics of why nectar is produced suddenly at a specific time of the day and what are the different hormones that control it. The composition of nectar is also something I'm very interested in. Sugars such as sucrose and glucose are predominantly the major constituents of nectar. But to call nectar a sugar solution would be a shame because nectar is actually a very chemically diverse liquid. It has amino acids such as proline and glutamine. It has free fatty acids. It has proteins in it, such as nectarines. So you can see here a mustard nectar that has been run on a protein gel shows multiple protein bands, suggesting that there are proteins with many functions. One of the proteins that we've already figured out the function for is actually that it is an antimicrobial protein. So stuff like this is coming up in the research as we look more into nectar. The other thing that is very rich in nectar is special metabolites, which is something that we are just starting to look at. And all these constituents are important for dictating the health of the pollinator because pollinators are known to forage on certain kinds of nectar and certain kinds of flowers. So to connect the chemistry of the nectar with the health of the pollinator is something that we are trying to understand as time goes by. This kind of overarchingly forms the basis for my lab in collaboration with my postdoctoral advisor, Dr. Clay Carter, where I trained on nectar biology. We are, as a group, trying to understand the different nectary genes that function to change the nectar chemistry, which then dictates how the pollinator health is affected by the chemicals in the nectar, which then eventually translates to how crops are pollinated and food security is attained. The other focus of my lab has been to understand the phenomena of colored nectar, which is rare but not few in the plant kingdom. Nesocoda mauritianus is one such starkly bright colored red nectar producing flower, which is found in the Indian Ocean island Mauritius. And it is endemic there, but thanks to the University of Minnesota's plant conservatory, we now have this plant growing in St. Paul in a protected space. So this allowed me to go and research the nectar of this plant. And the conservatory is an amazing place curated by Lisa Aston Philander, and it has an amazing staff of people who are always willing to help us look into cool plants. So if you ever want to work on plants, let me know. Now, this nectar was supposedly uh, um, pigmented due to this pigment right here, which was reported in 1998. But our research has shown something different. It's also interesting to note here that this plant is pollinated by an endemic gecko of Mauritius, Phelsuma. And as you can see here, a study showed that the Phelsuma geckos prefer red nectar over clear nectar, suggesting that the color has evolved to attract maybe the geckos. An undergraduate researcher working with me, Catherine Hall, one day as she was tracking the flowers in the conservatory, found that the newly opened flowers actually did not have blood red nectar. They had yellow nectar. And as time progressed over 24 hours, the nectar turned red. And this was fascinating for us because we thought that the nectar just starts off red. Biochemical analysis showed us that the pH of the yellow nectar was 6.5, and its spectral properties showed an absorbance at around 350. As the nectar progressed to an orange color, we found that a second peak appeared at 500, and the pH had increased to 7.6. And finally, the blood red nectar had a very different spectral property, and its pH was highly alkaline at around 8.7. Chemical studies and analysis by an undergraduate, Adam Kostanecki, and a professor, Adrian Hegeman, and his graduate student, Stephen Brockman, showed that the yellow coloration is formed primarily due to cinnapaldehyde, which then changes spectral properties as the pH progresses to an alkaline pH. And finally, the red peak, which we see here at around 500, is produced by a pigment, nesocodin, which is a novel pigment that we have discovered, and this has been patented by the University of Minnesota Twin Cities. And it's interesting because it has a stable imine bond, and also it is formed due to a condensation of cinnapaldehyde with the amino acid proline. If you remember, I had said that amino acids are found in nectar, and here we find a new function for the proline to give it a red coloration. This is actually synthetic cinnapaldehyde at a pH of 6.7, showing a yellow coloration, and then it's allowed to react with proline at an alkaline pH to form the red color. So we could actually form the red color in a test tube by just adding the constituents. Now, the nectar world is full of secrets such as these. There are exotic nectars that are found across the world that have different colors, and very little is known about them. So for example, Daubinia zaheri here has blue nectar, and Melianthus comosus here has black nectar. So this is something that we are starting to look into. And at St. Kate's, we will be working with students to biochemically characterize some of these pigments and also find out what enzymes are found in the nectar that catalyze some of these reactions. The secrets of nectar continue. There are so many species out there with so many questions. For example, Liatris here attracts monarch butterflies like crazy. Nobody knows why, what is in the nectar that causes such a specific attraction. So this is something also we'll be getting into in the future. With that, I would like to acknowledge the amazing team that I have always had the pleasure to work with. I also would specifically like to thank the undergraduate researchers that have worked with me over the years. I've learned a lot from them, and I hope to mentor and work alongside many more undergraduates at St. Kate's. With that, I would usually like to open the floor for questions, but if you have questions, please email me or My profile is up on the Senkate's website. Please email me with questions. If you're interested in working with me, I would really be interested in talking to you. Thank you for your time.